This protocol describes sample preparation and data reduction required for successful neutron spin echo studies of collective membrane fluctuations of relevance to biological functions and practical applications of lipid membranes. NSE directly accesses membrane dynamics on the nanosecond timescales of key membrane functions, with the unique advantage of isotope sensitivity for probing selective membrane features inaccessible with other techniques. Studies of membrane dynamics on the nanoscale are essential to understanding the molecular mechanisms underlying membrane properties and membrane-protein interactions implicated in various cell pathologies. The method provides researchers new to NSE with detailed guidelines on designing, preparing, and characterizing lipid vesicles for successful experiments and subsequent data reduction, analysis, and interpretation. Demonstrating the procedure will be Teshani, Kumarage, and Nguyen, a graduate student and an undergraduate student from the laboratory. Working inside a hood, prepare the lipid suspension by dissolving accurately weighed lipids in 1 milliliter of solvent with manual mixing. Dry the lipid solution by gently streaming an inert gas in the vial while slowly rotating it at an angle. To thoroughly remove the residual solvent, place the vials overnight in a vacuum oven at 35 degrees Celsius. On the next day, hydrate the lipid film with 2 milliliters of heavy water to obtain a lipid concentration of 50 milligrams per milliliter and vortex the hydrated lipid suspension until the lipid film is fully dissolved.